You can do it either here or with this, whichever one you want. No, just and on the screen you can see it anyway.
Hi everyone, thank you for being here. I think we'll go ahead and get started. I imagine folks will continue to roll in. Today's session is on biorepository and specimen management. Jeff Edberg is going to be presenting. He's the director of the biorepository and also a professor in the School of Medicine. And I will ask you to introduce yourself a little bit. He's great to work with. So when he does this presentation, if you ever need to call him, he's great to talk to. Now give him lots of work. Just so you know, the next session, the first Thursday of February, will be how to complete an HSP. It's going to be 2 hours long. We're going to start at 11 in the morning on that Thursday, finish at 1. We will serve lunch. We're going to have a restricted attendance. We're probably going to, you'll have to, we will send out the information, but you need to go online and then register. It will be that Eventbrite. Third Thursday, we're going to have Steve Yoder from the School of Business, and he's going to come and talk about negotiating and hopefully clinical budget, negotiating clinical trial budgets. We met with him. He does a lot of negotiation training, but he's in the School of Business, but he's not done clinical research. So we've met with him and kind of gave him the picture of what it takes to be a coordinator and a PI in negotiated budgets. So I think it'll be really helpful. He was very interesting to talk to. So I'm going to let Jeff go ahead and begin.
Thanks, Penny. So as Penny said, I'm Jeff Edberg. I'm actually in the Division of Rheumatology in the Department of Medicine. And the reason I got involved in this kind of work was through our own research in lupus, where about 15 years ago now, we got involved in some genetic studies where we needed to recruit thousands of patients and have thousands of DNA samples ready to be genotyped. And I thought, well, gee, how do I do that? I have no idea what I'm doing. So we had to build a biobank kind of on the fly to do that, and it worked well, and it allowed me to transition at the time to the GCRC and now the CCTS to provide some of our expertise to help others do similar types of things. So what I tried to do here was to pick some things that I think you might want to hear about, but certainly if there are other questions you have, just make it more of a discussion rather than a formal presentation, that's fine. There are a lot of potential issues we could talk about, and like I said, I may not have picked everything that you think is interesting, or there are other topics you want to hear about, let me know as we're going through, and I'll be happy to address them. So, and I'd really like to do is step back and have you think about what is it you're trying to do when you're having a new study. What do you need to collect? What are the goals of the study? You really need to understand what the scientific goals are, then back into what is it that I need to collect, and how do I store it? How do I handle it? Where does it go? All those kinds of questions. And perhaps one of the first important issues to think about is when you're thinking about biospecimens is how are they going to be used? Are they used clinically? Does that mean that you're going to have reportable results generated from a blood tube that's drawn? That's going to influence patient care in some way, shape, or form. Or is this really for research use? Or is it for both? And those are important because you might handle them differently. So samples are going to be intended for clinically actionable findings. They've got to be handled in a certain way. That's not just the research way. Not that research is sloppy or bad. It's just not quite as rigid as a clinically actionable finding result would be. In research, as you know, you are flexible and you understand what you're doing, and you can change how you do things depending on experimental need. But when you're doing something that's clinically actionable, it has to be done the same way every time, irrespective of how you think it should be done. It has to be done the way that the protocol calls for. Currently, at UAB, what we do for most studies is you can have something that needs to be clinically actionable. That blood tube just goes to directed outreach, and they do what they do. We get research tubes in our setting and process them. Then they should back up. So within the CCTS, we do have a couple laboratories, and I'll get to that in a little bit. But one of the labs that we have for handling research specimens is in the CRU phase 1 clinic on JP15. And so we can handle all your specimen processing needs. We can store specimens, short-term, long-term, or ship them, whatever needs to happen. But one of the things we oftentimes do is help. Well, this tube goes to outreach, so that has to get tubed down outreach. We don't touch that. Versus kind of the issue of research where you're getting extra tubes that are going to facilitate the research that you're trying to do in your patients. So just because it's clinical use doesn't mean we can't touch it. Our lab is in the process of getting CAP certification. We're not really CLIA certified because we're not testing anything. We're a research processing facility. So we'll be CAP certified for biorepository specimen handling functions. but there won't be any tests that we're necessarily performing that would allow us to be CLIA certified, at least at this point. So when you think about biospecimens, it's important to remember that just a tube of blood or plasma or whatever it is, DNA from it, that's great, but you've got to know something about that sample. And there's increasing interest in how does one link, particularly data from electronic medical record, which we'll touch on at the end, to specimens. But certainly at a minimum, you've got to make sure you understand the demographic data associated with the specimen, certainly any clinical phenotypic data, high level about the specimen. The ultimate holy grail is to get it linked to the EMR so you can pull in any data that's available for your study to allow you to better utilize those biospecimens. And then certainly any research-based results that might be associated with it. When you're running a biobank, typically you're not putting data on the specimen, but you just need to be aware of how do I link my data, whether it's data from the EMR, other clinical data, or research data with the specimen that's stored somewhere. And we have some solutions for that I can show you as we get near the end. You need to think about the types of specimens that you're banking. Certainly healthy versus disease-associated materials. If you're collecting blood, if you're working with patients, sometimes blood counts are quite different in some patient populations. You just need to be aware of what it is you're collecting and how you want to use your samples to know what you can or can't. You need to think about whether you're going through tissue versus liquid type specimens. And liquid can be a range of things, but typically blood, urine, CSF, saliva are the types of things we typically see, but there might be other types of specimens too. And then there's increasing interest in the collection of stool for microbiome research. And each of these types of specimens have distinct collection, handling, and storage needs. And there are solutions at UAB to help you with all of these different types of specimens. So what I've tried to outline here, this isn't, I'm sure there are others on campus who do things like this too, but these are at least 3 resources to start with. If you're looking at liquid type specimens, typically our lab, the CCTS, can handle those. I'll give them a link and you'll see my contact information. Again, we're certainly happy to talk. You don't have to work with us, but you can come to us for help if you want. That's fine. We don't charge anything for that. We're happy to help you think through what it is you're trying to do and how you might get some advice on how you might do it. We're happy to help you do it too, if that's what you need. For tissues, there are a lot of issues with tissue collection, processing, and storage that are very complicated and depends on the type of tissue you're working on. We don't do that. There's a very good group in the pathology department, also supported by the campus center, Dr. Grusel's group. He's on the 4th floor of the Ziegler building, and Kathy Sexton runs that facility. They're very good at both curing tissue from the OR. They also have a small biobank of bank tissues that's available. Might be helpful. I've given the website there. For stool collection and analysis, UAB's put a lot of money into the microbiome resource core and a microbiome center. The particular core for handling stool samples is run by Dr. Casey Morrow in cell biology, and I've given a pretty long, obnoxious link there for that too. But you just search for UAB microbiome resource and you can find their information. And as I said again, each type of specimen has very different types of handling and collection needs, and so you need to consult with appropriate people to make sure that you're doing things the right way up front before you get to the end of your study. Because I just had somebody come to me yesterday, they wanted to do RNA analysis, and they haven't collected specimens in a way that will allow that. The study's done now, so they can't do it. That's a shame, because if they just would have talked to us, we could have suggested ahead of time how they could have perhaps set things up a little differently to help them anticipate that need. So again, things to think about before you start collecting specimens, and then not even necessarily for a biorepository, just for even for a study. You've got to think about how are your specimens going to be useful for the research that you're trying to do, and that you need to think about how and when and where you're going to collect, how are the samples handled and processed, and where is that happening, how are you going to store the samples, how are they going to be retrieved, and how do they get to where they're supposed to go to be analyzed. Again, we can help you think through those things. We've worked through all of these stages of studies many, many times. We don't charge, and I'll show you the link at the end. We have a monthly clinic that you're welcome to come to and ask questions without any obligation to use our services. You also need to think about what kind of assays your specimens are going to be used in. And certainly the easiest part is the protocol will dictate we're going to measure X, Y, and Z or whatever it is. But you also need to think a little bit about what are possible future needs. And technology is always changing, and you can't always anticipate everything. But there are some ways to think about simple ways of adding some storage specimens to allow you to do other types of studies. There was another study where there was a lot of money invested in collecting plasma for analysis of circulating DNA, and they only collected enough to do it that one time, but now there's a whole new technology that can be applied to that, and the study, again, the samples are gone. And that was a shame, because they very simply could have just added a little bit more at collection time, and they could have had enough to do it again with new technology. So it's always important to think not just about what I'm doing today, but what are potential future needs that might come up, or opportunities that might come up. Okay, so any questions so far? So what I'd like to do is kind of walk through the life cycle of a research specimen, at least from our perspective, going from specimen collection all the way up through the utilization of the specimen. So when you're collecting specimens, first and foremost, you need to think about your informed consent. This isn't a talk about IRB, and I'm not an expert in that, and I won't pretend to, but you do need to think about how you set up your informed consent. You have to think about what's the participant consenting to in the study. What will research findings be shared? Will incidental findings of potential clinical use be shared? Those all are issues you've got to go through in your human subjects protocol. You need to think about how your specimen is going to be used in research, and that needs to be articulated at some level in your HSP. But there's an art form in your human subject protocol that you don't want to be too restrictive. You don't want to say, I'm going to use it to measure X, and then leave it at that, because then, as I say, things change, technology changes, opportunities change, and maybe you want to measure Y now. And if you limited yourself to X, you're not going to be able to do that without consenting people. So you need to keep your language sufficiently specific, but not so specific that you eliminate possible other uses. You also need to think about how samples might be used for future research needs. And that's always been a somewhat sticky area in IRB, because we don't know what the future is going to be. You can't say in a consent form, well, I don't know what I'm going to do with your sample, but just let me do anything you want if you want. But there are ways that you can word your human subjects protocol and your consent form to allow for future use. That could be future use within your study, for other collaborative or ancillary studies. And actually just yesterday, many of you may know, the common rule is being revised by Health and Human Services, and preliminary rules were posted yesterday that you can go to, and I've given a link at the bottom here. And in particular, one of the sticking points was the issue of how does one think about using samples in the future after they're collected, particularly if they're de-identified, but also potentially identifiable too. And the new rules, I think, are going to be much easier to work with in the context of future use than the old rules were. This is not federal statute yet, and who knows what's going to happen after tomorrow. But there's good news that these guidelines, as they've just been posted now, will make it a little more straightforward in how to set up samples for future use in both the consenting process and in managing the IRB, the interaction with the IRB for future studies. You need to think about what it is you want to do with your samples, right, obviously. So if you're trying to measure something, you need to know what you need to collect, and you need to know particularly time temperature requirements. A lot of samples are temperature sensitive, or a lot of analytes are temperature sensitive, some are time sensitive. So you need to know about what it is you're trying to do with your samples. Again, we can help you, and a lot of other people on campus who can help you with these things too. I'd urge you to always reach out. Sometimes it's not always clear what's supposed to happen. You get a lot of studies that come to us where they have no idea how they're supposed to handle these things. We don't always know either, but we at least know where we can look and who we can ask and try to help you work through those issues. You need to think about what types, so I'm going to focus on blood here, obviously with tissues and stool. Those are completely different sets of expectations and issues, and we're not expert in those. So I'm going to really focus on blood, which is what we deal with every day. You need to think about what type of blood do you need to collect. There are a lot of different types of vacuum hairs, and I'll just go through those briefly so you can kind of get a sense of how we think about this. But you need to understand, so what's happening with this blood sample? Am I interested in the liquid part, plasma serum? Am I interested in nucleic acids, DNA, RNA? That can be both cell-associated, and more recently there's increasing appreciation for the role of circulating DNA and potentially RNA as biomarkers or prognostic indicators of disease processes. And each of these types of nucleic acids have different collection and handling needs. Or sometimes you need cells. So you need to understand what it is that's going to happen with the cells, with the blood, and also be anticipatory. How might these be used in the future to help further our studies? So when you come down to what kind of blood tube do I collect, it can be a little dizzying sometimes. BD has a nice blood collection guide with different colors, describes what's in there. I got the link on the bottom there. I think you've got all the slides for you to read that. If not, then CC Jess can share these slides and see these blood links. But this is a good resource to help you understand what the different types of blood tubes are. So I picked a couple different types to kind of give examples of how you might think about different blood tubes. So if you're interested in actually analyzing cells, generally you're going to want to make sure that your blood is anticoagulated so it doesn't clot. Because when it clots, the cells are going to get all bound up in that clot that forms, and they're not going to be very useful for analysis. But the type of anticoagulant can make a big difference in cell function and cell viability. So for example, purple tops or EDTA tubes, if one is interested in B cells, which make antibodies, EDTA tends to kill B cells, so that's not going to be very good if you're going to have to go to a B cell lab and want them to do something. They're not going to get very good results. Time and temperature are important. Cells are not going to last forever sitting around on the vacuum. Sometimes you want to put them at 4 degrees, but again, B cells, if they put at 4 degrees, they tend not to be very happy. None of this is absolute. It's not like you can't put it in a refrigerator and have nothing, but if you're interested in B cells, it's typically better not to refrigerate with ice. But if you're interested in T cells, it might be better. So you really need to understand and have a sense of what it is you're trying to go for and what it is you're trying to do with these cells. And again, just emphasizing time of processing is important, particularly for cells. Cells are always changing and responding, even when they're drawn into a vacuum. Sometimes people like to use CPT tubes if you're interested in isolating peripheral blood mononuclear cells. Those are tubes that have a separation medium in them. You just collect it, mix it, throw it in the centrifuge, and 20 minutes later you'll have your PCAMS. If you do it manually, it takes a little bit longer, a little bit cheaper, just depends on what your requirements are. But these are all important considerations. A lot of people are interested in measuring RNA, looking at a transcriptome. You may have heard about looking at transcriptomics. That's by measuring RNA, where you measure the transcript levels for different proteins. That's a very dynamic process, and the transcriptome is always changing. And as cells are collected, even if it's for a couple hours, those cells are changing, the transcriptome is changing. So you have to, if you know you're going after transcriptome analysis, you've got to get those cells isolated and stabilized in a way that will preserve the RNA for future use quickly. The other thing to think about is blood is a complex tissue, right? There's a lot of different kinds of cells in blood, and again, depending on what you're looking at, you may or may not need or want to separate out different types of cells. We can do some cell separations, but there are a lot of good cores on campus that can do much more detailed cell separations. You can use flow cytometry to isolate out very small cell subsets using flow cytometry. And again, we can help you think through those things if you need help with that. But these are all things you just need to be thinking about as you design your study. Sometimes you're interested in plasma serum. Remember, that's the liquid part. So serum, by definition, is the liquid that's left over after the blood clots. And plasma is the liquid part that remains when the blood does not clot. Some things that you might want to measure in blood are going to be much more sensitive in serum, some in plasma. And there are guidelines, actually, I didn't put the link here, I'm sorry, but the clinical pathology lab has a very nice chart which has all the analytes that they can measure in clinical pathology with the type of specimen that's required, plasma serum, and typically which type of vacuum tube you want to use. If that's particularly important when it's plasma, because remember, when it's plasma, you have all different kinds of anticoagulants that can influence potential measurements downstream. And again, as always, time and temperature are important. And there are many, particularly inflammatory cytokines that can be very sensitive to temperature and time. You know, the plasma is stored within an hour, so you're going to lose the ability to measure important mediators. So here are a couple of different examples of serum collection tubes. So remember, serum clots, so there's usually no additives in the tube. Some tubes nowadays have what's called a clot enhancer to help facilitate clot formation. When you collect the serum tube, you've got to make sure you collect it, that it's mixed, and that it sits for 20 or 30 minutes before you spin it down to allow the clot completely. Some tubes have a gel barrier. You can see the tube on the right there with that little yellow thing at the bottom of the tube. That's actually like a lax plug, and what that does is it allows you to separate that clot from the liquid part very cleanly, so you can maximize your harvest of serum. If you just put it into a regular tube, you can still get most of the serum, but the serum tends to kind of mix with the clot. You don't get a clean interface there. So that gel barrier is good to allow you to get better yield of serum. But there may be some analytes that don't like the gel separator, so you just need to be aware of what are you trying to do. Typically these tubes are red top or tiger tops, but sometimes they're gold or red. They're just a variety of different types of tubes that you can use for serum. Plasma, again, these have anticoagulants to prevent clotting. There are a lot of different types of plasma collections. We've only shown a couple here. The typical ones are green, purple, lavender, and yellow. And again, like serum, some plasma collection tubes have a gel barrier, like the tube I showed there. Again, the reason for that is just to facilitate the collection of the plasma, so you get a really clean separation between the cells and the liquid part. When you just stick it in a regular tube, you always have to leave a little bit of plasma behind because it's sitting right on top of the cells. You don't want to get any mixing that occurs there. Each anticoagulant has a different property. Green tops are heparin, lavender is EDTA, yellow is ACV. And you just need to be aware of what you're doing with the sample because you need to collect it in the right tube. I'll give an example here of why that's important. So if you're interested in nucleic acid, that's DNA, RNA. Typically, we've always thought of that as being white cell associated. Red cells don't have nucleic acid, don't have any DNA. RNA, but the white cells that are circulating in your blood are the ones that contain DNA. Typically, DNA is the same from cell to cell unless you have somatic mutations that can occur in cancer. So if you're interested in DNA, if you go with whole blood, it doesn't really matter what your cell type is. But it is important what kind of tube you use. And for example, those green tops that contain heparin, certainly you can get cells from that and you can get DNA from that. The problem is that heparin is a very sticky molecule that sticks to DNA and it co-purifies. Heparin is a very nice inhibitor of the enzyme that's used in the PCR reaction, which in almost all applications of all DNA, PCR is used. So if you're using heparinized tube, it's likely that your DNA will not be compatible with PCR. So if you're interested in DNA, we typically try to go for the purple top best, sometimes yellow tops work fine. RNA, so remember DNA encodes all your genes. It's transcribed into RNA, and the RNA is translated into protein. So RNA can give you a measure of how much expression is occurring within a particular cell. The issue with RNA is it's very susceptible to degradation. There are RNases which chop up RNA. They're everywhere. And if you don't handle your sample in special ways, your RNA will degrade. It just happens. You cannot stop that unless you do something to actively intervene. Typically, if people are interested in RNA, there's a special type of vacuum cleaner called a tax gene tube that has a stabilizer in it. It's a big tube, but it only collects 3 mils because there's a lot of the stabilizer in there. The issue with those tubes are expensive. You can only buy them in batches of 100, and they expire usually within 6 to 12 months. So it's kind of obnoxious, but they work. You can also use something called RNA later, where we would typically isolate cells from the blood to concentrate them, and then you add this RNA later solution, which lyses the cells and then activates the kinases. So you can put them in the freezer and keep them for use later. But if you just take blood and you throw it in the freezer, or you try to separate cells and throw it in the freezer, your RNA is going to be degraded. It's not going to work. So I just emphasize that because the study that came to me a couple days ago about RNA, they had just put cells in the freezer thinking, well, that's fine. We'll figure out what to do later. And so now 2 years into it, that's what they got. So they can't do it in RNA work. The issue with RNA is, again, remember, blood is a complex mixture of different types of cells, and every cell is different. And part of the reason they're different is they express different genes, and so that's reflected in the RNA transcriptome. So it's just important to think through if your study wants to look at RNA transcript levels or whole genome transcriptome analysis, where your RNA comes from is actually important. As all of us, our blood counts vary over time, right? So proportions from cells vary. That means that if you take whole blood and you isolate RNA, the level of RNA is going to vary from gene to gene depending on how many of the different types of cells you have. So sometimes people like to separate out cells and then get RNA from separated cells. It really depends on what you're trying to do. you can get into enormous expenses. We're trying to get RNA from cell subsets, but sometimes that's what you have to do, depending on where you're trying to measure your study. Okay, so any questions about collection for cells, isoserum, nucleic acid? So let's talk a little bit about labeling, transport, and processing. Obviously, hopefully this is all common sense, but I just want to reiterate because we are amazed sometimes we still get things that we get labels that are illegible. We can't read anything on it, or it's all smudged, or the label rips off. It's important that samples are labeled properly. It's important that you think about how our samples are getting from the point of collection to where they're supposed to go. Typically, patients seen in a clinic, that's great, and I know TKC, the hospital lab, is very good about helping and drawing research tubes. But then you think about who's going to get them, who's going to transport them to our group, or to wherever they're going. Again, time's an important factor there, right? Because they're just sitting somewhere waiting to be picked up. You need to make sure that that's being addressed in an adequate way. And then specimen processing. You need to know how are these samples supposed to be processed. Again, we can help you with that. That's hopefully the current theme here, that we're here to help you. You don't have to use us. If you don't want to, that's fine. But we're here to help, provide guidance. And then certainly you need to think about, if you're not using your samples right away, how and where you're going to store them. And I'll go over a couple different storage strategies. There are a lot of different kind of freezers, different temperatures you can store them at. A lot of that may depend on how you're going to use them and when you're going to use them, because freeze thaws are not, those are important biological events. When plasma is frozen and then thawed and then frozen again, things change. Some of the proteins will denature, that won't be available for detection anymore after that first freeze thaw. Other proteins like antibodies, you can freeze thaw a number of times, and that's fine. Just depends on what you want to do with it. But again, just to be aware of that it's a variable that you need to know about. So here's an example, the purple top on the top here, you see with the label kind of ripped off. We couldn't see most of the ID. We encourage the use of pre-printed labels if possible. We can help with that if you need help with that. But that way there's no handwriting, they tend to stick better. And we have labels that are really good that don't come off. If the tube is put on ice, sometimes labels come off. We have labels that don't come off in ice. And they're paper labels, they get wet. You can't read them very well after that. So you just need to be careful and think about those things. So why is specimen management important? Well, obviously your results are only be as good as your strategy was for collecting, processing, and storing your samples. You need to make sure that everything is done appropriately for your study to allow you to get the endpoints that you think you're interested in. Quality specimen management is required, and it's an active process, not something just kind of do on the fly. You really need to be thinking about this. Like I said, there are resources on campus to help with this. Ours certainly is available. But we have a lot of experience with this. It's not that we don't make mistakes. Everyone makes mistakes, and we'll screw up sometimes too. But quality management really is an active process, and we put a lot of time and thought into that about how do we do this the right way for each study. So how can you manage your specimens in your study? So first of all, you got to know your protocol. And I know sometimes when the lab manual has particular industry sponsored studies, they tend to be these huge things. It's like, oh my God, how to figure this out? And we have problems too, because it seems sometimes what you need to say in sentences they put into a book. And what you really need to know is buried in there somewhere, but you just need to find it. But it's really important to simply know your protocol. Again, we're here to help you wade through that. We've seen a lot of these lab manuals, particularly for industry studies, and we can help you try to find out what it is you're supposed to do. You need to know where they're going. Are they going to stay here? Are they going to Provence? Are they going to hospital lab? We can help with shipping if you need help with that. Our group is IATA trained and certified, so we can help you with that. And you need to know what's going to happen to your samples. What are your expectations of where they're going, so you know that you're getting back what you think you need. And again, the sensitivity. Sometimes samples are sensitive to light, temperature, and time, so you need to be just aware of those things. Another thing you need to know about, particularly for industry studies, where they need to have standardization across all sites. How are your samples processed? And typically where we get the most confusion is how are they centrifuged? And when we think about centrifugation, there's time, how long are they spun, what temperature are they spun at, and how fast are they spun. Those are the three variables you need to think about. The time and temperature is pretty straightforward. You just need to know what that is. And certainly in our lab, we will adapt to whatever the protocol requires. The difference in clinical pathology is they try to help with research sometimes too, but they don't have the flexibility. They've got to do things the way they do it because they're running clinical samples all the time. We will do it the way that your study wants it done. Where things sometimes get a little confusing is the issue of centrifugation speed. And there's the issue, there are two ways of expressing how fast the rotor spins. You can do RPMs, revolution per minute, or you can do G-force. And there's a formula to convert this to, and people always get confused when they're given a number. What does that mean? And then sometimes centrifuges don't make it easy also to know how to convert or which one you're looking at. The most important thing is just to recognize there are two different ways of expressing speed, G-forces and RPM. Typically it'll say 300 times G or 300 RPM. Those mean very different things in terms of the speed of the rotor. We can help you with that. There's a simple formula that you can convert the two. It depends on the radius of the rotor. So every centrifuge is different. That's the other point here. The RPMs determine what the G-force is, and the G-force is directly proportional to the radius of the rotor. So the bigger the rotor, the more G-force you can have as you go out in the centrifuge. So you need to know these things as you're trying to do it yourself, which is fine. You just need to understand that you need to know what G-force is in your centrifuge based on the radius of the rotor. So I mentioned labeling. And we really encourage reprinted labels whenever possible. Sometimes that's not. If it's not, make sure that the labels are clearly written so that wherever they're going, they can be read. When we aliquot specimens out of the blood tube, for that we always like to use pre-printed labels because they're going to a freezer. They're going to get wet with frost. We want to make sure it's a pre-printed label that's not going to come off the freezer. We like to encourage use of barcodes, so you have an independent identifier on that sample that's easily identified by a scanner, and you don't have human error in terms of data entry. As I said, the labels have to be appropriate for storage conditions. We're storing cells, those are going to be in a cryogenic freezer. That's at minus 190 C. Not all labels will withstand that kind of temperature. We can help you understand what kind of labels you need based on the freezing requirements. And the other issue is going back to the labeling. We've got to have an ID when the sample is received, because once we create an aliquot, a label on that sample, when we freeze it, it's very hard to relabel in frozen because it's wet now. The label's not going to stick. So it's really important to have everything lined up before the sample's processed. You have the right ID and what other information you're in the label, because it's really hard to change that label after it's frozen. So I mentioned there are different types of storage conditions. Typically, you think of three. There's kind of minus 20 C storage. That's like your household freezer is at minus 20, basically. Most freezers are frost-free, right? That is really nice at home, but that's a disaster for your samples. The reason it's frost-free is it warms up and cools down on a cycle every day. It's warming up and cooling down. That's really bad for your samples. So if you've got a frost-free freezer, you don't want to be using it in the lab. You can still store at minus 20, and you can find some non-cycling minus 20 freezers. But you have to be careful. It's frost-free. Don't use it. Typically, minus 20 freezers, we really think of just for short-term storage of some samples, plasma, serum, DNA, but it's not as stable as the better long-term minus 70, minus 80 storage. Those are typical freezers like you see on the top there, the big laboratory freezers. Those are always non-cycling. That's why the frost builds up in them. That's a pain, but there's a reason for that. That way they're not warming up and warming your samples. And typically for minus 70, minus 80 long-term storage, that's where you're going to put plasma serum, glucose, DNA, sometimes blood, storable blood. And they're usually, those specimens are usually pretty stable under those conditions. It's important to think, even though minus 80 is cold, samples are still evolving over time, even at minus 80. So time is always an important issue. You need to know if you're measuring something with plasma serum that's just really sensitive, you probably don't want to store it for a long time. You probably want to use it as quickly as possible. Last thing is cryogenic freezers. These use nitrogen typically to store, to provide cooling. This is used most typically for storage of cells, and cells can be actually cryopreserved, so stored and frozen in a certain way that allows the cells to maintain viability when they're thawed. That's a somewhat more complicated process. You just don't throw them in the freezer. You got to freeze them in a specific way where it's rate controlled over time before they're put into liquid nitrogen. Our CCTS facility has a bank of cryogenic freezers that are automatically filled, so you don't have to worry about liquid nitrogen levels. But some of you may see little dewars that sit in the lab that you manually fills them every month or so. That's fine too, as long as you monitor and maintain those. There are some what are so-called mechanical freezers. These have compressors that actually pull it down to minus 150, 160 C for storage of cells. But typically, the easiest and the best solution for storing cells is a cryogenic freezer that uses liquid nitrogen. They're expensive to run and maintain, so we've got some of the CCTS that we need help with that. We're there to help, and we can store things there. So just because you have a freezer, that's great, but you got to make sure that you know what's in the freezer and where things are. And there's some pictures there of how freezers can look in labs where things are just shoved in. It's all iced up. Nothing's labeled. You don't know where anything is. And unfortunately, a lot of research labs, that's the case. We don't do things like that. Our freezers are totally organized. They have racks with boxes in them. Each box has a grid with samples in the grid, so we can map where every single sample is in our freezer at all times in an inventory database I'll show you in a second. So these are just things to think about. Sometimes it's just not sufficient to throw them into the freezer. I've actually literally seen one study where they were putting their vac containers into styrofoam cups in the clinic, carrying them over the lab, and just throwing them in the freezer, and when you open the freezer up, with all the styrofoam cups with tubes in them, and that was a disaster. You know, again, the labeling wasn't good. You couldn't read most of them. Some of the tubes would crack. And the other thing I forgot to put in here, I should point out, be very careful with glass vac containers. Glass will break because water expands and it freezes. And we actually had a case where an investigator had collected vac containers and they froze them and they wanted to get DNA, and the tubes cracked. You had cracked blood tubes everywhere. It was just a disaster. So be very careful when you're storing vac containers. If they're plastic, usually it's okay, but if it's glass, I would not recommend storing this. You need to get those transferred somewhere else. The other thing is don't put glass tubes into those styrofoam holders that you sometimes get tubes shipped in because it constrains the tube. It'll crack if it freezes in that. So be really careful. If you are going to freeze a tube, put it on the wire rack so it's got some room to expand. So what are your options when you need to freeze your samples? Freezers are expensive. Typical minus 80 might be $10,000 or more. A cryogenic freezer is going to be even more than that, plus the cost of liquid nitrogen. And there's also the issue of the energy consumption. Right? Every time you plug in a minus 80 freezer, it's a huge drain of energy. I've been told the Shelby building is by far the worst energy hog on campus because of all the minus 80 freezers in that building. It's so big. So what are your options? You're doing a study and you've just got a couple boxes, you need to buy a freezer. Fine, if you want to keep your own samples, maybe that's what you want to do, but there are other options. In the CCTS, we've always had the ability to have store samples. Typically they're the ones we've worked up, not samples that come in from things that are already worked up. But starting just this year now, we've been able to expand, and it's not quite finished yet. We're going to have a new biorepository facility with bank freezers where we can talk about accepting samples to store, so you don't have to buy your own freezer. Again, we don't necessarily have to work them up for you, but we will have to have certain requirements that when they come into our freezer, we're going to have an inventory. So they're going to be labeled properly. We'll have to know what's there and things like that. But there's a resource on campus where we can help you store things, so you don't have to buy your own freezer. And what we've done is we bought, there's a new technology actually in freezers, believe it or not. Most freezers, as you know, like your household refrigerator freezers, use a compressor to cool the air inside the freezer. This new company called Stirling, they use something called a Stirling engine, which is a different type. It's not a compressor, and it doesn't use any of the oils and gases that a compressor would require. And it's a very reliable, very small. You can see in this freezer, that's a picture of one of our freezers actually. This little engine is in that top part, so you have a lot more room available for storage. The advantage of these is they're very reliable. All the cooling on the space shuttle on the International Space Station uses these Stirling engines. They're very energy efficient. It uses about a quarter of electricity of a standard ultra-low freezer that you see in the lab. It generates very little heat, so you don't need the cooling requirements. It's all natural refrigerants. So these are really neat freezers. They're a little bit more expensive, but we've got 8 of them now. So we've got a lot of room to help people store things. So you have to buy your own freezer and have all the associated expense of energy utilization of that. This will be in the Shelby building. Other issues to think about in terms of freezer management, and we put a lot of thought and time into thinking about this. We need to make sure that the freezers are maintained and monitored. We have a system where our freezers have dual temperature monitoring with 2 independent probes. We have 24/7 monitoring. We do one remote monitoring as a web-based application using a wireless system called Temp Tracker. It's an independent probe that transmits temperatures every 15 minutes to a server. You can log at any time from anywhere and see what's happening with the freezers. All the freezers are on backup power. So a lot of issues when you think about your freezers. You just don't plug it in and walk away and done. You need to really think about these issues if you have your own freezer, how you're going to make sure the temperature is always right, what happens on the weekend, when it goes down, how you can get notified. We've got all that thought out. We have solutions in place to help us with that. You also need to make sure that staff handling the samples in the freezer are appropriately trained. All of our folks are trained in good lab technique. They've gone through all their appropriate training, bloodborne pathogen, life safety, GCP, IATA for shipping. As I mentioned, we're in the process of being accredited by CAP for the handling and storage of specimens, which will ensure that we're reaching the highest quality and are using all the appropriate techniques. So, as I mentioned, you need to know where your samples are. A lot of labs have paper logs, spreadsheets. That's fine, but it's really not the best way to do things. You need to think about how to manage your specimen inventory, and the issues to think about is how do you track the location. Our samples barcoded, and if so, how do you track those barcodes? You need an audit trail to know when a sample goes in, when it comes out, how many times it's thawed, those kind of things. You need to be able to track the full specimen life cycle, so when's it received, when's it stored, when's it taken out, shipped, when does it come back, all those kinds of things. You need to report on that, and you need to be able to import into the database when you import the samples. In the CCTS, we have a very robust system to do all this for us. It's called Encore. Most of you probably heard about Encore now. That's going to be the clinical trials management system used here on campus for all clinical trials. But it also has a specimen management system built into it. It's web-based. It can track all those issues I pointed out on the top there. It can easily generate barcodes on the whole acre samples. It can generate shipping logs. It can do some full specimen tracking. One of the beauties of Encore is that if you're using our lab to help with your specimen management, if your sample is being run through Encore as a clinical trial, your patients will already be in there, and we can just come in on top of that and utilize those existing records to add the specimens to those patients without any additional work. So that's a big advantage of using Encore. Just here's some screenshots. This is actually from Encore. You can easily see this is a particular patient, and in this case there are 3 different times when we receive samples on different dates. That's the case level, and then underneath each case there are different specimens that we can see on Encore. And behind each of those specimens is all that information I mentioned. When was it received, where is it, what happened to it, who did it, all that kind of information. We can also track when specimen derivatives are made. So if you have a plasma sample and you want to sub-aliquot a small amount of it, that can be tracked when it happened, where it happened, and what happened to the sample after it got sub-aliquoted. That's all trackable within the system. It can also generate reports. So this is just a simple storage report. So if your samples are in a particular box, we can generate a report with a single button to show you where all your samples are. You can confirm that's there and tell us which you need to pull, ship, or whatever. You want to do it on your own. Encore is probably a bit much for an individual lab, but there are other software packages you can try to manage your own freezer. FreezerWorks is probably the standard for individual labs for managing their own freezers. It can run standalone on a PC. There's also a network version if you want that option. It's a very robust solution. What it doesn't do, though, is link to clinical trials like Encore, because that's not what Encore. Another one's called Freezer Pro. I've not had any experience with that, but it's one I know others have used. Open Specimen is another option. Many of you remember CA Big. That was the Cancer Institute's attempt to try to formalize the collection of data and distribution of data. Didn't really go very far, but one of the components of CA Big was CA Tissue, which was meant to be a freezer management, sample management application. The NIH abandoned that, but it got picked up by a company in India, and they have an open source policy where you can download the code for Open Specimen, and for a fee, they'll help support you if you want to do that. And then there are also laboratory information management systems. Those are typically very expensive, many tens of thousands of dollars, but those work well too. As I said, options that are typically not good are paper records and Excel spreadsheets. They can just get you into trouble. So you really need to think about inventory. And again, we can help you with that if you need some advice. So the ultimate holy grail, right, is you got all the specimens, how do I link data to them? And obviously through research results, that's research labs got to figure out how you're going to do that. What Encore can do is we can certainly track the dates, times, and locations, and who pulled specimens and put them away. But the real issue is how do you get clinical data associated particularly with patient samples. And typically, you'll think about storing your clinical data somewhere other than your specimen inventory, because they're really very different types of data, but you need some way of linking them. The medical record number in the past has kind of been a taboo, but that's really become the standard now, and I'll show you one way that is being done both in UAB and elsewhere around the country about how to link specimens that are stored somewhere to real live data in the electronic record. And this really is the future of good biorepositories. There's a lot of money put into collecting samples, storing them, using them, and to make maximal utilization of that investment, you need to be able to get to real data that's changing over time. And that's where the EMR comes into play. The ability to link those two things is really the future. And the way we do it here, and a lot of academic institutions do it, is with an application called I2B2. This was developed with an NIH grant awarded to a partner system in Boston. And the idea was to basically take a snapshot of the kind of medical record, put it into a separate place, and then allow that to be queried to ask questions: how many patients are there? Sort of like cohort identification. If I want to do a study and I need patients with certain criteria, how many potential patients are even here at our medical center? And this is a way that you can do this without any identifying information being shown. You can just quickly query the system and find out how many potential patients that might be available to you. Likewise, and we've done this now, this is a live example where in Encore, where we have medical records for patients with particularly, this is for one of my studies in lupus, there's nothing identifiable here, but what we did was we put the MRNs that we had DNAs on into this I2B2 system, and then I did a simple query: how many active lupus patients are there at UAB that have a DNA in the freezer in our biorepository? And the answer was 36. Again, it doesn't tell me who it is. That's only knowable to the study. It happened to be me in this case, but the advantage of a system like this is if you were interested, someone else was interested, and said, gee, I really would like to get some DNA. Who should I talk to? Is there anybody here who even has this? I2B2 can tell you, well, somebody has it. The people who run I2B2 can be the odds broker and look up who's got these samples in our users that you can then talk to about forming a collaboration. These are not samples that are available to anybody else other than the study. When you give us samples, they're your samples or nobody else's, and nobody else can take them. But what we try to do here is to help people find points of collaboration. And if there are samples already collected for something that might help your study, we can hook you up with who has those samples, and you can negotiate whether those samples can be shared or not. Obviously, sharing of samples is a sensitive issue. You need to make sure that consent is done properly and that they're available to be shared. And that's not always going to be the case. But at least with tools like this, we can at least find the potential for working together. And in fact, next week at the same time over in the Shelby building, Jim Sanina, who runs the Informatics group at the Informatics Institute, is going to talk about I2B2. And certainly at the first level, I2B2 really is cohort identification for patients, but the next level is to merge in specimen data. I mean, we've got specimens stored in freezers, and hopefully he'll mention that in his talk too when he talks about I2B2. So what can you do? As I mentioned, the first Wednesday of every month right here, we have consultation clinics, and no appointments necessary. You just come in with any question you have about clinical study, translational study. It doesn't have to be about specimens. There'll be somebody here. There'll be biostats people here, some from the nutrition unit, typically some from the nursing unit, and some from my group. You can come with any question, and we can kind of help you. Benefit is lunch is provided, so if you come with questions, you get a free lunch. It's the first huge month. But we're here to help. And again, coming to the clinic doesn't obligate you to use the services of the CCTS in any way. It's really just to help you design and implement your study. Here's some contact information for our group. There's my email and phone number. As I mentioned, we have a couple of different labs. Our primary lab for clinical trials is in Jeffery Tower. I'm on 1515. That's on the CRU phase 1 unit. It's actually a new lab. We just moved there a couple months ago. So it's nice now. Some of you may remember the old lab. It was pretty dingy looking. The new one's really nice. It's much bigger. We have a lot more space. We have some freezers up there for short-term storage that we can help you if you need to store some samples for a couple of weeks or months before they go out to where they're supposed to go. There's a phone number. We have a lab in the Shelby building. I have multiple labs in the Shelby building. The main lab is in room 230, and that's where we can do more specialized work. That's where we do DNA extraction, cell isolation, cell cryopreservation. And that's also where we do the long-term biobanking. That's where all our long-term storage freezers are. We have -80 freezers and cryogenic freezers, and many of those now we're moving down to our freezer room, which will be in the basement of Shelby, which gives us a lot more space, more specimens in the future. And then some of those web links at the bottom, those are just some general links that we've found useful over the years. Actually, OHS, the Occupational Health and Safety, has a lot of good guidelines, safety-related issues, IATA for issues related to specimen shipping. And everyone, I hope you all know that if you ship biospecimens, you have to take the IATA course and get certified before you can ship biospecimens. That's actually important. And dry ice is a separate issue too. You need to make sure you understand that you're trained to ship dry ice before you do that. This Edberg organization was actually a group called the International Society for Biological and Environmental Repositories. They have a lot of good information about how to manage specimens, how to build a biorepository. It's a wealth of information. And the last one is it's a commercial website, but it's all about sample handling. They have a much more comprehensive guide about the different types of vac containers and when and why they might be used. That's a good supplement to the Becky Dickinson website that was in one of the earlier slides. Okay, any questions?
Is it possible that if you do a study and you do not process the samples at the CRU lab or the Shelby, can you store them in the system?
Yeah, so that's what we can do now. Now we have expanded freezer space. We can start to talk about that. Obviously, before we take anything on, we're going to want to make sure that they've been handled appropriately and labeled appropriately, and we'll need to know how they're labeled so we can integrate them into our inventory system. This is all contingent on getting them into our inventory system. So
there is a submission for that?
I'm sorry?
There is an application for that?
Yeah, so the room is, so that freezer room in the Shelby basement isn't finished yet. That's a whole other saga with UAV construction and the high construction project. How fun that can be. It's supposed to be done about 6 months ago. We're still fussing. So we still need to work out how this is going to work. I think in talking with Dr. Kimberly, he's very interested in seeing if we can get MRNs on samples. Again, samples belong to the investigator, not anybody else, and nothing is shared outside of just identifying that a sample exists with a particular phenotype, and we can be honest for people. So if you put patients in with some sort of renal disorder and somebody else is interested in that, they're your samples. No one else is getting them until they talk to you and form formal collaboration. But there might be incentives that if you're willing to have your samples available for discovery like that, there might be incentives, financial incentives to help facilitate that. And we need to work that out still. But as always, I think, as you've hopefully realized, our fees are pretty low and reasonable, I hope. And we'll have the same philosophy here. The CCTS really helps us keep those prices low by helping kind of provide that infrastructure. Thank you.
So the Encore system that we're going to be moving the clinical trials to, right, will that specimen management piece of that be available to others as well? Or?
So we've not worked that out yet. It's a little complicated given the way that Encore is set up. It's not clear yet how or if that's going to be possible. We have such a large presence in Encore already. We've got about 500,000 specimen records that we've already put in there in the last couple of years. And because we work with studies across campus, we're already going to kind of be in there. It's just not clear how we're going to kind of segregate out. So if your lab wanted to have their freezer in there, it's just not clear yet how that would be set up. That's not something the company that wrote Encore had really anticipated when they first set it up. So we're working with them to kind of talk that through and see how there might be ways to do that. Because the issue right now is there's not a way to isolate out, so you can see your freezer but not mine, and vice versa. Label printing, so all the labels we make are done through Encore. There's not a way to keep my printers in my group and your printers in your group. There aren't ways to kind of segregate those things yet. So we'll need to work on that. Other questions? Thanks. We still have those cells in your group.
That we were talking about the lipid nitrogen. No, we're not ready for that yet. Like you said, it was a preliminary discussion, but we just want to make sure.
Well, but there's another side that we actually have cells. Right?
Oh, I'm thinking of one.
Right, there's another one. There's another one that we already have. Right, yeah.
Oh, for training. Yes.
Yeah.
And so we've had some screen failures, unfortunately. So
Well, they're still there. We got another next I don't know. It's been
yeah, we worked.
Yeah, they did, except where they would, we would hold them until we injected them. It's been a, I think usually.
Commissions are not numbers, but commissions are free, just like Jill. And if she has any questions, Jill is her resource.
Yeah. We do
We
check.
can check with the coordinator and Tracy.
And Jill
I'm
is
working
already
temperature.
collaborating with Keisha, which we can
They,
put
yeah.
them training off.
Yeah, I think they. From the company side, I think they're organization wide, I'm sure they planned it on their, I think, destination wise. Right.
No, I have about
Have it on
one, but I think she has another.
Yep. So.
That's not taking a lot of space, just fine.
Share it, I'll make sure that you can get information.
Hopefully, we can.
That's what I was, and I choose, and I know for sure.
Yeah, the coordinator for, if whoever Sarah called.
Right, and I actually did that.
But I don't have any.
And,
Okay.
and Sarah can get assigned right now.
It came in this
And
big
it said the pre-screen line.
Yeah, like the whole hand, yeah.
Thank you.
So, but yeah, I think she's so much. But she's been, we have one that we're shooting now, so I'm hoping that we'll, hopefully we'll get one soon, but. Mhm. Thank you.
Get ready for it, she'll have time for it. And we really do it maybe the next one. People want us to watch what they're going to It is very time consuming. That's why I like to questions. Never know what you're doing. We get outside of that's one of the, that's how they're recreational answer to the patient question or they're ready because that's like, yeah, new patients that are visitors, it's easier. Yeah, that's right. They say that way. Thanks, Jeffery.
Did you hear from him?
You're going.